Hi everyone, I am Dr. Rashni Verma and I am going to take a quick revision session on the chapter Cell, the Unit of Life. This chapter is from class 11th Biology, and in this video we are going to discuss some important points for your NEET examination 2019 examination. In this slide I have summarized all the important points which has came or which can come for the next year examination. If you are preparing for medical entrance and if you have done your studying part, from these slides you can prepare more or you can get to know if these points you are knowing that it means this topic is completed for your NEET 2019. Okay, without wasting any time, let's get started. So, discovery of cell. When the cell was discovered. But before that, we have to study the definition of the cell. Cell. What is cell? Cell is a structural and functional unit of life. Then next question comes in your mind that who discovered the cell? Robert Hooke was a scientist who was working on the cork tissue, and then he saw that tissue under the microscope. Then he saw some structures like this kind of structure which he has drawn. It was looking like this, and he has named them. Later on, he has named those structure as individual structure as cells. But it was a dead cell. Then later on, Anton von Leeuwenhoek was a scientist who discovered a living cell. Nucleus was discovered by Robert Brown. Then comes the cell theory. Cell theory was given by Sheldon and Saquon, two scientists, Sheldon and Saquon. The cell theory consists of 2 points that time. All the living organisms are made up of cells and all the functions are the product or functions of cells. Means all the organism, whatever is there, all the living organisms are made up of cells. And all the functions or whatever it is produced, it is done by the cells. Then this theory is modified by a scientist, Rudolf Virchow, and he has modified and named that theory as a modern cell theory. That modern cell theory states that they have clubbed 2 points. All the living organisms are made up of cells. All the functions are the product or functions of cells. But he has given one additional point that all cells arise from pre-existing cells. Means one line of cell is formed, then these line of cell produce some other cells. Okay, in his language he has stated omnis cellula acellula. Okay, then smallest cells is mycoplasma or PPLO. PPLO is pleuropneumonia-like organism. Pleuropneumonia-like organism. Ostrich egg is the largest cell. And example of unicellular organism are amoeba, paramecium, euglena, chlamydomonas. There are many few, but I have listed some of them. And multicellular, there are so many examples. All the higher plants and animals comes under multicellular organism. Then there are type of cell, basically two types of cells, prokaryotic and eukaryotic. Prokaryotic means the cell having a primitive nucleus, a very basic nucleus. Means it is not having, the nucleus is not having a proper nuclear membrane. Okay, it has a very basic nucleus. And we have studied 5 kingdom classification, as you remember. Monera, Protista, Fungi, Animalia, Plantae. So from those 5 kingdom classification, the kingdom Monera comes under prokaryotic cells. Prokaryotic cells are the cells which are found in the members of Monera kingdom. And most common example of Monera kingdom, all the bacterias. So bacteria are having prokaryotic cells. Then becomes In a prokaryotic cell, no membrane-bound structures are present. So, membrane-bound structure are known as organelles. So, no organelle is present in a prokaryotic cells. Here, we'll study the structure of a bacterial cell, which is example of prokaryotic cell. So, this is a bacterial cell. So, there is no membrane-bound structure. But one structure is there, and which is membrane bound also, that is ribosomes. Ribosomes are present in prokaryotic as well as eukaryotic cells. Then some bacteria, not every bacteria, but some bacteria is having a additional layer which is known as glycocalyx. Okay. Then this is a gelatinous layer, glycocalyx, and if this layer is present like a loose sheath, then it is called as slime layer or slim layer. And if this capsule, that glycocalyx layer, it is very tough. In some bacteria it is loose and it is very tough in some bacteria, then this is known as capsule. Okay. And then comes cell wall is present. Cell wall is made up of peptidoglycan. And peptidoglycan is the membrane which decide the bacteria is gram positive or gram negative. I'll discuss in detail later on. But till now you should know this thing, that cell wall is having peptidoglycan. Then the cytoplasm is having all the enzymes necessary for all the processes. So all the enzymes are present in the cytoplasm. It has a genetic material in the form of DNA, which is circular DNA. The DNA is circular. And the plasma membrane, which you can see here, it has certain enfoldings inside the cytoplasm, and these enfoldings are known as mesosomes. So please do remember this structure, mesosome. There are some enfoldings in the plasma membrane which are known as mesosomes, which contain respiratory enzymes. Respiratory enzymes are used in the production of energy in the bacteria. So these enzymes are present in mesosomes. And the ribosomes, which is present in bacterial cell or prokaryotic cell, it is 70S ribosomes. 70S ribosomes. Then plasmid. Plasmid is also a kind of genetic material. It is a circular extracellular genetic material which help in the transfer of genetic material from one cell to another. By this structure, the bacteria transfer its infection or the genetic material. It is very important in the division role, in the division of the cell, the plasmid. And plasmid have a very crucial role in biotechnology, which will be discussed in detail in the chapter biotechnology. I'll tell you in detail, but till now you just remember plasmid also play a very crucial role in antibiotic resistance. Then flagella. Flagella consist of filament, hook and basal body which help here. Flagella helps in motility. By this flagella, the bacteria moves from one place to another. Okay, then comes the fimbriae. These are same structure. See, flagella in motility and fimbriae. Fimbriae are different structure. These are projections on the plasma membrane which helps in adhesion of the bacteria to some place, which helps in adhesion. I was telling you about the peptidoglycan. The cell wall has a protein called peptidoglycan, and we know gram positive and gram negative. So when we treat with the stain, gram stain, the bacteria, if In some bacteria, this peptidoglycan is present, and in some bacteria, this peptidoglycan is not present. In the bacteria, the peptidoglycan is present is known as gram-positive bacteria, and the bacteria in which peptidoglycan is not present is known as gram-negative bacteria. So, which is if the peptidoglycan is present, they will take the stain and they will stain in purple color, that is gram positive. And if do not take the gram stain, that is colorless, the cell will appear colorless, that is gram negative bacteria. Eukaryotic cells, all the protists, ants, animalia and fungi, all the rest four kingdoms comes under eukaryotic cells. Okay. Animal cells is having a plasma membrane. Plasma membrane is made up of proteins and phospholipids. This is the plasma membrane. This membrane is plasma membrane. Nucleus is a double membrane structure. There is nuclear membrane, and inside the nucleus is a structure called nucleolus. And on nuclear membrane, some pores are there. They are called nuclear pores. And some thread-like structure are there inside the nucleus, which are known as chromatin fibers. So, cytoplasm is there. In the cytoplasm, so many structures are there. Organelles, membrane-bound structures are there. So, first we'll come to ER, endoplasmic reticulum. Two types. Endoplasmic reticulum are of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. If the ribosomes is present on the endoplasmic reticulum, then it is known as rough endoplasmic reticulum. And if it is not present, then smooth endoplasmic reticulum. Then Golgi apparatus is there. And then Golgi apparatus के बाद centrosome. Centrosome is made up of centrioles and pericentriolar fiber, and this centrosome is not present in prokaryotic cells. Okay, centrosome are made up of centrioles and pericentriolar fibers. Why I have highlighted this in red? Because we are studying an animal cell here. So this structure is not present in the plant cell. Rest of the structure are same as animal cell. Mitochondria, lysosomes, vacuoles, cytoskeletal structure like microtubules and microfilament. These are the structure. You just get to know the names, and later on we'll study in detail every structure. In plant cell, we have cell wall. Cell wall is made up of cellulose in plant. Plasma membrane, it has additional is a cell wall. Plasma membrane is the same as like animal cell, but it has an additional layer called cell wall. And then vacuole. In animal cell, there are vacuole many and small. In plant cell, it is a large vacuole and it is a single vacuole which contains sap and minerals. Nucleus is double membrane, and nuclear membrane nucleus is like same as the animal cell. Double membrane, nuclear pores, nucleolus is filled with nucleoplasm, thread-like structures. Golgi body is same as in plant cell. We also Golgi body is also known as dictyosome. Mitochondria is the same. ER the same. SER, RER. Lysosome is the same as animal cell. Ribosomes is the same. There are two additional structures which are found in plant cell are plastids and microbodies, which we will discuss later on in detail as an individual structure when we will discuss the organelles. So, after discussing the plant cell, we'll discuss the cell wall in detail, only those points which are important for examination. So, cell wall is present in fungi and plants, in both the organism, fungi and plants. And cell wall give the shape to the cell, and it protect from the mechanical damage and function. And cell wall is selectively permeable, which means it allow only certain molecules to pass across it. Okay. And in algae, the cell wall is made up of cellulose, galactans, mannans, and calcium carbonate. This point you have to remember because they can ask like what is the composition of cell wall of algae, or one of the example they will give and they will ask. Okay, this is present in the cell wall of algae or plant like this. And plant also cellulose is same. Cellulose, hemicellulose, pectins and proteins. Cell wall is made up of in plant cellulose, hemicellulose, pectins and protein. Plasma membrane. Plasma membrane is a common structure which is found in animal cell as well as in plant cell. So, before studying the structure of plasma membrane, we should know there are some models which came to give an outline of the structure of the plasma membrane. So, there are basically two models we have to study for examination, sandwich model and fluid mosaic model. So, first we'll study sandwich model. Sandwich model is given by Davison and Daniel. Davison and Daniel. Okay. Which state that cell membrane is made up of bilayer of phospholipid sandwiched between alpha globular protein layer. So what does it mean? We'll see here. See, this is the layer of phospholipid layer, and this is two layers. That's why we call it phospholipid bilayer. Okay, this is a phospholipid head and this is the tail. This is the phospholipid head and this is the tail. Head is hydrophobic in nature and tail is hydrophilic in nature. Okay, and this is the protein layer. So this model states that two layer of phospholipids is sandwiched between the protein layer. Why this model was rejected? Because the non-polar protein portion would separate the polar portion of the phospholipids from water, causing the bilayer to dissolve and makes the structure unstable. That's why the structure was rejected. Then fluid mosaic model was given. It states that plasma membrane is made up of proteins and phospholipids, the same line, and it also states that phospholipid is a bilayer, consists of head, tail, same structure it states, but this is variable. It has a variation as compared to the sandwich model, that position of the protein as discussed in sandwich model like this. It states that proteins molecules are embedded in the fluid matrix of lipid bilayer like this. And the proteins which are there are alpha globulin protein, and there are two types of protein on the basis of position, that is extrinsic protein, intrinsic protein, or partially or completely embedded in the phospholipid layer. Okay, intrinsic protein. Extrinsic is lied over the surface. Okay, and these proteins makes channel for transfer of molecule, but it does not allow all the molecule to pass. That is the reason the cell membrane is semi-permeable in nature. Those protein channel allow some molecules to pass through and it inhibits some molecules to pass through. That's why the nature of the cell membrane is semi-permeable in nature. So guys, if you like this video, please like the video, and if you have any doubt or you want any modification for this video, then please let us know in the comment section, and please do subscribe for the upcoming videos. You will get notified. Thank you so much.